consciousness exists as a fluid spectrum of measurable bioelectrical frequencies. These neuronal networks operate throughout the neuroaxis, driving distinct states of engagement or rest. This polygraph tracing tracks the 3 strict parameters required to define the sleep-wakefulness cycle, or SWC. We monitor cortical electrical activity through an EEG, muscle tone through an EMG, and ocular movement through an EOG. During non-REM sleep, muscle tone decreases, eyes remain still, and the brain's electrical activity shifts into slow, synchronized delta waves. Rapid eye movement, or REM sleep, creates a physiological paradox. The muscle tone readout flatlines entirely, indicating complete atonia. Yet simultaneously, the brain and eye readouts spike into high-frequency, low-voltage patterns resembling active wakefulness. Identifying how the brain coordinates these 3 distinct signals requires locating the internal generators driving them. Mapping the functional anatomy of the cycle requires isolating individual components within the neuroaxis. By sequestering specific nodes, researchers can observe how the remaining network responds to the loss of a specific signal. Before the invention of non-invasive functional imaging, mapping a distributed, overlapping neuronal network posed a severe mechanical challenge. The primary investigative methodology of the mid-20th century relied on surgical exclusion. Researchers used systematic brainstem transections, physical cuts across the neural pathways, and highly localized tissue destruction via diathermocoagulation. This diagram maps the continuous neuroaxis of the cat brain, extending from the spinal cord up into the cerebrum. By severing at these precise diagonal coordinates, researchers permanently isolated structures above the cut from those below it. If a behavior or electrical pattern disappears in the isolated upper cerebrum after a specific cut, the generator for that signal must physically reside below the line of transection. By systematically moving these transections centripetally, starting at the spinal cord and moving upward through the brainstem, we can reverse engineer the exact anatomical origins of both sleep and wakefulness. We begin the deduction at the absolute base of the central nervous system, the transition point between the spinal cord and the medulla oblongata. Look exactly at transection line D. This marks a complete separation at the C1 spinal segment. It creates what is known as the encephalae-isole preparation, cleanly severing all somatosensory input originating from the body. The physiological question is direct. If you remove ascending sensory impulses from the peripheral nervous system, can the isolated cranial machinery still cycle through sleep and wakefulness on its own? This top EEG tracing shows the result. Even entirely disconnected from the spine, the encephalae-isole maintains a normal, alternating sequence of synchronized sleep and wake-like electrical activation. Contrast that with the tracing below, taken from a cerveau-isole, a higher intercollicular section. Here, the isolated cerebrum exhibits constant high-voltage slow waves and fixed pupillary constriction. It is permanently trapped in a state of synchronized sleep. While peripheral nerves and spinal cord impulses conduct sensory data that can modulate our sleep, like a comfortable temperature or a quiet room, their complete removal does not stop the cycle. The core biological machinery required to actively generate both sleep and wakefulness resides entirely within the cranial vault. Isolating the brain raised another major hypothesis. Is sleep a passive response to fatigue metabolites accumulating in the bloodstream? This schematic illustrates a radical 1976 experiment. Researchers grafted an isolated encephalae-isole dog head directly into the circulatory system of an intact host dog. They shared the exact same blood supply, but their neural pathways remained entirely separate. The intact host dog maintained a standard circadian rhythm, characterized by wakefulness during the day and consolidated periods of sleep at night. These side-by-side pie charts compare the results. The outer ring shows the host animal's normal circadian variance. The inner ring represents the implanted head. Despite receiving the exact same blood chemistry, the isolated head exhibited a monotonous, even alternation of sleep and wake phases, unaffected by the time of day or the host's state. The isolated head lacked crucial neural timing signals from severed vagal and carotid nerve receptors, which normally relay state-modulating impulses to the brainstem. The chemistry of the blood does not dictate the architecture of the cycle on its own. Hardwired neural inputs are strictly required. To find those hardwired inputs, researchers moved their transections progressively higher, probing the pontine structures of the brainstem. Here on our anatomical map, transection line B isolates the medulla and the lower pons from the upper midbrain and the cerebral cortex. This is the mid-pontine pre-trigeminal transection. This diagram compares the normal electrical cycle against the resulting isolation. Without signals from the lower brainstem, the cerebrum never enters synchronized sleep. The electrical activity flattens into the low-voltage, high-frequency pattern of persistent wakefulness. The isolated upper brain exhibits continuous ocular alertness, and the characteristic slow delta waves of NREM sleep entirely disappear from the polygraph tracings. Severing the lower brainstem traps the upper brain in wakefulness, indicating that those lower structures must actively emit the ascending signals required to induce sleep. Gross transections proved the general location. To pinpoint the exact cellular hubs driving the sleep state, investigators shifted to localized tissue destruction using diathermocoagulation. They targeted small, highly specific clusters of neurons within the caudal pontine tegmentum. These coronal cross-sections map the specific locations of those targeted lesions. Look closely at the dotted overlay areas mapping the caudal pontine reticular nucleus. Notice the dotted regions versus the striped ones. When these precise dotted areas were destroyed, researchers recorded a statistically significant permanent decrease in both NREM and REM sleep. The physical engine of sleep had been broken. Neuropharmacology verified the mechanism. By microinjecting carbachol, a cholinergic agonist, directly into this exact same pocket, researchers could artificially force an increase in synchronized EEG sleep in an otherwise awake subject. The caudal pontine reticular nucleus serves as an active engine that forces the brain into the sleep state. How does a small, localized cluster of neurons in the lower pons physically enforce electrical synchronization across the vast expanse of the cerebral cortex? This technical schematic tracks the physical wiring. By utilizing anterograde degeneration studies, researchers mapped the exact path of the neural fibers radiating upward from the damaged caudal pontine region. Moving to panels C and D, we see the targets of this hypnogenic projection. The degenerated fibers fan out bilaterally, reaching deep into the midbrain reticular formation, the thalamus, and the lateral hypothalamus. These ascending pathways split and cross over, ensuring that the sleep signal is broadcast equally and simultaneously to both cerebral hemispheres. The infrastructure of sleep operates as a massive, upward-radiating neural broadcast system, projecting synchronization outward from a dense brainstem core. This schematic visualizes the true nature of the sleep-wakefulness cycle. It is a constant dynamic struggle between two opposing neural networks built into the neuroaxis. The reticular activating system in the upper brainstem acts as a perpetual engine, constantly driving the cortex toward the high-frequency state of wakefulness. Watch this network interaction. Sleep initiates when the lower pontine brainstem actively overpowers that waking drive, sending out ascending hypnogenic impulses that throttle the activating structures and enforce slow-wave synchronization. Consciousness represents a spatial dialogue between distinct anatomical structures, mapped and proven through the meticulous logic of the neurophysiologist's scalpel.